Hey, Bio 1 students, Mr. Yachts. We're going to continue on with our Chapter 8 notes, which deals with protein synthesis and DNA. And I went over with you already in notes how DNA is the structure of DNA, a little history of how it was discovered with Watson and Crick back in the 1953s. Basically, that's where we're going to pick up today. I want to talk a little bit about, as you see on the screen, replication of DNA. So please take notes, put squares around and underline things and highlight what's in red, just like we did the other day in notes. So replication of DNA, a little introduction. Really important that you highlight, circle, put an asterisk beside it, whatever you need to do. We're going to build an exact copy of DNA in DNA replication, okay? Later on, we're going to talk about a process where we build a near copy. That's different, okay? This is no different than me going to the copy machine, taking an original, and running 100 copies of that original off and having 100 copies of the same information. That's what we're doing with replication, okay? This process occurs, if you remember from last chapter, this is going to occur in the S phase of interphase. It doesn't matter if we're talking about mitosis or meiosis. We got to make a copy. So when I go from that parent cell to those two daughter cells, I'm going to get the same information in both of those daughter cells. So it makes sense that I got to make a copy so when I get two cells, they both have the same information. Now, this can be just making blood, skin, muscle, bone cells that keep us alive every day. That's mitosis. Or if I'm talking about that happening in an egg or sperm cell, now I'm passing that code from one generation to the next generation because we're talking about offspring, right? So this replication of DNA is extremely important regardless if I'm talking about making more cells or I'm talking about making more people, right? Or dogs or cats or whatever you want to talk about. All right. Now, I'm going to try and make this really simple. If you take my Bio 2 class someday, I go in a lot more detail with this, but I'm going to break it down into 3 steps. And we're going to make a foldable in class starting on Monday, and we'll start putting this information in that foldable. So right now, I just want you to highlight some of these steps, and eventually we will put it in foldable. Okay, so we'll just get this out of the way today, and then I'll tell you what we're going to do with this information on Monday. All right, so 3 steps. Okay, the first thing that happens is the DNA has an enzyme called helicase that's going to come down and literally break the bonds of DNA. Okay, now this is an interesting process. Let me show you a little diagram, and you guys have a collar sheet. You might want to write this stuff in on that collar sheet. Mine's already done. So we have this helicase. Write that in. It's going to come right up from the bottom and basically go right down the center of DNA. So think of a ladder. So say I have a wooden ladder and I have the steps or the rungs of the ladder. It'd be like you taking a chainsaw and cutting the middle of each of those steps. And I have half the ladder goes this way and half of the ladder goes the other way. Okay? We have a name for that. Those are called parent strands. So as I cut that in half, these 2 halves are called parent strands, and that's what I'm going to build off of, okay? Now, this may sound kind of odd to say this, but these bonds that hold the bases together, AT and GC, we talked about this in class. AT and GC are held together by weak hydrogen bonds. They're easy to break. So helicase is an enzyme that just comes up through here and is going to break these bonds in the middle, okay? So I have a G go this way, C go this way. A goes this way, T goes this way. So we're just going to separate that ladder into 2 equal halves. Hopefully that makes sense. So that has to occur first, because if that doesn't occur, then we're going to be in trouble. Okay, so that's the first thing that has to happen. So Helicase has to break those bonds. I don't know where this went, so let me kind of get this fixed here real quick. All right, so that's the first thing that has to happen is DNA literally unzips. So it's just going to break right down the center, and we're going to unwrap or unzip that DNA, and we call that a parent strand. Hopefully that makes sense so far. All right. Step 2. Okay. Since we have this ladder exposed now, so I got an A over here, a T over here, I have a G and a C, or vice versa. I have these little pieces of DNA that are floating around in the cytoplasm. We talked in notes already about how a nucleotide is a sugar, a base, and a phosphate. Remember that term, okay? So what we have happening here is we basically have, I don't know why my link's not working. It must have it messed up here a little bit. So I just have these little pieces kind of floating around here, and they're going to find their complementary match. Okay, what that means is, let's say, kind of follow along with me. This is where it gets a little confusing. Okay, so I have this exposed parent strand. You can see it on my screen here. I'll highlight it with my mouse here. We have this basically exposed strands that are just kind of circling around or circling through, and there's an exposed G, there's an exposed A. On this side, the opposite, T's are exposed, okay? These little pieces, you see them over here, they're floating around. There's a phosphate, a deoxyribose in this case, and guanine, adenine, cytosine. There should be a thymine floating around somewhere. And they're just waiting. And as soon as I unzip that DNA, they're going to find their complementary base. So if I have an A exposed, T is going to hook on. If I have a C exposed, G is going to hook on. And this is happening on both sides, okay? And this is called a daughter strand. It's the new strand I'm starting to form. So I have a template and I have a new strand on both sides that this is going on with, okay? Now, what happens then is we're just going to start building a new DNA strand on this side and a new strand on this side. We're making a copy. Imagine that, okay? So what's going to happen then is I am making 2 complete sets of DNA on both sides. Now, follow along with me here. Look what happens, right? So if you look at the diagram, right? So if you go back and we look at this, then you're going to notice something, okay? Look at the 2 sides. You guys got a color sheet. You can look on this, okay? Look at the 2 sides. I have, as soon as this little thing disappears at the bottom, I have a GC at the bottom. On the other side, I have a GC, TA, AT, CG. Both of these sides are exactly the same information. I am making a copy on both sides. So when I get done, those 2 DNA strands are going to separate in mitosis or meiosis, and I'm going to have the same information that's going to go into both cells. Okay? So hopefully that makes sense. It's pretty simple. DNA just splits in half. I build new copies on both sides, and I'm going to get not just a close copy, not a near copy. I'm going to get an exact copy of DNA on both sides of my diagram there, okay? And that's going to happen every time a cell divides. So hopefully that makes sense. I'll review this a little bit on Monday, and we're going to put it in our foldable anyway. So that is called replication. 3 steps, DNA unzips. The bases are going to find their matches, and I'm going to make 2 exact copies of DNA on both sides. All right. Now I just want to introduce this next thing. I might as well finish this slide up, and then you'll put this in your notes. We'll put this in our foldable eventually, and we'll call that good. Okay. I want to talk a little bit about RNA. So I'm going to change gears. We're done with replication. I just kind of want to introduce this. RNA, here's the problem we have. The problem we have with RNA is that, or the problem we have with DNA, I should say, is it stays in the nucleus. It never leaves. Now, I talked about ribosomes in the cell chapter and how ribosomes make proteins, blood, skin, muscle, whatever we need. The problem is DNA can't leave the nucleus. It's stuck in there. So how am I going to make a protein from a blueprint that's in a different location than where I'm making the protein, right? For example, let's say, and I had this problem when I built my house years ago. I got the logs, the building materials came out of the UP of Michigan, but my blueprint came from Wisconsin. So I had to get the blueprint that was in a different location and the building materials was in a different location and get the two together where I built my house. You got to have the two in the same place, okay? So since DNA can't move, how do I do that? We use RNA. So think of RNA as being like a taxi that's able to take the DNA blueprint to build proteins and take them to the building site, okay? Now, that is called transcription. A little different. Look at the difference here. This is building a near copy. So we're not making DNA out of DNA. We're making RNA from DNA. So it's going to be a close, near copy, but not exact. So kind of keep that in mind, okay? And there's really three ways that DNA differs from RNA, okay? So let's just get this slide in and one more thing, and then we'll call it good. The sugar is different. The name is different, RNA, DNA. Ribose is the sugar of RNA. Deoxyribose is the sugar of DNA. This is a good test question someday. DNA has three bases that are same as RNA. ACG are the same, but uracil is put where thymine is at in DNA. So that's a swap. So the base thymine is replaced by uracil and RNA. It still bonds with A. ATAU does the same thing, but it's a little different that way. And the third difference, and then we're going to call it good, I'm not going to go through this whole process today, is RNA is a single strand, DNA is a double strand. So RNA is not a double helix like DNA is. It's just a single strand. You'll see why. I'll explain that later. All right? So hopefully that makes sense. What I'd like for you to do is get this information, copy it down in your note packet that I gave you, highlight, put the squares around, underline stuff, and then Monday, or if this is down the road another year, whatever day it is, may not be Monday in the future, but we're going to put it in our foldables. Okay? So, hey, have a great day, and we will talk to you later. See ya. Bye.